30 years ago, as it turns out. And some experiences that I had 30 years ago. I was older than you. I'm about 40 years older than you, but 30 years ago, I wasn't that much older than you. I was 24. And there was a group of people. I was at the University of Washington studying zoology. And the University of Washington the main campus is in Seattle, but they have branch campuses all over the place. And one of the places that they have branch campuses is on the San Juan Islands. Has anybody ever been to the San Juans? Really beautiful. You have? Where did you go? Did you go to Orcas? Do you remember?
I don't remember.
Okay. It's really neat. It's a really neat place, like this small group of islands. You have to get to them by boat. So we had to take the ferry out. And I knew a couple of other people who were applying, but I didn't know them well, and I didn't really know, like, we all had to apply. There was, like, you had to, like, get recommendations. It was something where you couldn't just decide you were going to go and then go. Instead, we had to apply and then get accepted, and I got accepted. And so I remember getting on the ferry, loading up my 1983 Honda Accord hatchback and getting on the ferry, and then I got on the ferry, and I did see at least one other person that I knew. I think I just saw one other person, I think, maybe two. And so we kind of arrived at this place, and that was, in April of 1994, so it was 30 years ago. And I didn't know anybody well. I knew a couple of faces and a couple of names, and that was it. But it was a really cool experience. We were there to do science. We were at the branch campus. There is a laboratory called Friday Harbor Labs. That's the name of the town. And what they study is marine science. So they have like boats there, and they have all sorts of research scientists that are doing stuff, and they teach classes. And they teach a variety of classes. The classes that my little group took, there were about 20 of us, were a marine zoology and a marine botany class. Marine zoology focused on invertebrates, so we were looking at like squids and sea cucumbers and echinoderms and all sorts of neat sea critters, the kinds of things mostly that you'd find in tide pools, because that's where we were able to go. So it was really easy for us to go visit tide pools and collect organisms, and we looked at things in tanks, and so crabs and starfish and chitons and limpets and mussels and barnacles and all that stuff is what we were looking at in the marine zoology class. And then marine botany, as it turns out, isn't really botany, because the things that are green that photosynthesize in the ocean aren't plants, they're algae, but we used to think they were plants, like not 30 years ago, but way before then we thought they were plants, and so that kind of stuck. So we call it marine botany, but really it's marine I don't know what you would call it, the study of algae, whatever that is. Anyway, those were our two courses, and we studied all those things, and we learned a lot. It was really interesting. There's, to this day, stuff that I remember. I still have like my old books and my drawings and all my notes and how about that stuff. But what was really interesting, and I'm not gonna talk too much about this, because while it's by far the most important thing, it's also not directly relevant to what we're doing here at school, but it's something that, like, my professional goals all have to do with you learning, but my personal goals all have to do with you being happy down the road. I want you to be happy in 5 and 10 and 20 and 40 and 60 years. I want you to live lives of contentment and happiness. And so I will just say that one of the things that we've found out about happiness is that universally, across age groups, across demographic groups, across cultural groups, gender, sexuality, all of these things, every single study, every single research project has shown that there's really one key piece to human happiness, and it's the quality of your relationships with other people. That has been determined beyond any doubt. And it's funny because much of the time we emphasize other things, and especially here at school, we're like, stop talking, stop texting, and stop like having fun with your friends, but actually that's the most important thing. It is the most important thing. It's just that in order to be a functional, happy adult with really good personal relationships, you need to be able to set those relationships aside and focus on other things. Because to be an adult, you have to like have a job and interact with community members, and maybe you're going to be a parent, and you have to figure out, you have to know how to do all that stuff. So that's kind of the nuts and bolts stuff that we're working on here in school. Like, you can't have friends if you can't get a job. Having said that, the friends are the more important part of that equation. So what's really interesting on a personal note, this group of people and I stayed in touch. Many of us, when we were only there for 3 months, but many of us came back to Seattle. So there was a group of people that sort of stayed in Seattle, and we hung out. You know, we would see each other. Some of us worked together. Like this one guy, Sean, got a job, and then they were looking for other people to hire, and so he was like, "Ooh, I know some people," and like 3 other members of that group ended up working for that organization. And some of us lived together. Like we'd be like roommates, because we, you know, at that point we knew each other pretty well, and we basically lived together, so we knew we could do it. There were relationships. A couple of people got married. Well, they started dating, and then now they're married. And but we stayed in touch, and then, you know, there'd be times where we like kind of drift out of touch, but then, you know, we get together. Somebody would get married, or there would be, you know, we would just gather for whatever reason. And this has gone on for 30 years. And it was 30 years ago that we went to Friday Harbor. So this year, we decided to gather at Friday Harbor. We went back to the San Juan Islands over Memorial Day, and this is our group. There were a few people who couldn't make it. There were people with other commitments. There were, you know, this one woman lives in Finland, like she's not going to be able to make it. And then there's additions, right? Because people have spouses and partners and kids and stuff. But this is like primarily a group of people. These people here are all people who have known each other for 30 years and stayed friends. And I will throw out, and this is the personal piece, I will throw out to you that that's a pretty unusual thing. Like, most people do not have a group of friends that they're still close to from 30 years ago. And if I could wish one thing for every single person in this classroom, it would be to have a group of people like this. Because this is a group that these people have stuck by me in my absolute lowest moments, and I've stuck by them in their lowest moments. These are people who I could go for years without talking to, and then I could pick up the phone and call or get together for a cup of coffee and instantly be right back where we left off in the spring of 1994. It's like, this is a worthy target. Just, you know, like to me, I feel like what I wish is that I wish I could say, like, here's what I did and here are all the awesome things, but mostly what it boils down to is I'm just incredibly fortunate, like incredibly lucky to have this group of people and have stayed in touch with them for all these years. So that's the personal piece. Where it comes back to the school thing is while we were there, we discovered some stuff, and I had forgotten, quite honestly, about a lot of this. It turns out that we all did research projects while we were there. Totally forgot that. But one of the people, there's a library on there, and one of the people found our book. I didn't even know this existed. They took all our research papers, everything, including like the photos and stuff, and they put them in a bound book. So this was our group, the Marine Botany and Zoology class, spring of 1994, and all of our work is in that book. And I looked through it, and what really struck me that's relevant to you all and relevant to what we're going to do for the last few weeks of school is how awesome it was. Like, we were not that much older than you. I was 24, but I was the oldest person there. Most of the rest of these people were 20, 21, 22. That woman was only 19. She was like still in her teens while we were there. She was really like on your level. And yes, we had taken a bunch of classes. We were all in our last year of college studying science. you are in your first or second year of high school studying a bunch of stuff. So I get that we were academically in a place that you're not yet, but in terms of what we were capable of doing, absolutely no higher level than you. We were held to a standard here that was really inspiring. We were told, you're gonna do awesome work, and nothing less than awesome work is acceptable. So that's what we did. And I'm gonna show you the work that I did. And I'm not gonna walk you through it or anything like that. I'll just show you a couple of pages of it just so you can see. So we took this book off the shelf. We looked at everybody's research projects. Some of them were looking at creosote on dock pilings. So dock pilings are the big pieces of wood that they sink into the shoreline, and then they attach the dock to that. But to keep those things from just rotting away, they put a chemical on it called creosote. It's like a tar, sort of. And so this one woman was like, well, I wonder if that's bad for the creatures around the dock. So she was looking at that. And some people were looking at marine organisms in tide pools. And what I did is I worked with a woman, not one of the ones who was there, but we were looking at algae. So what we wanted to do, and we checked with the instructor, we were like, look, one of the things that's been hard for us this quarter has been that if we want to learn these algae, we have to go collect them. And it would be really great if we had preserved specimens that we could look at to help us remember what these things are. And so our professor was like, yes, that would be great, go do it. So we did. Our project was to collect an herbarium, and this is what we did. I'm not gonna read this to you, or it's on Canvas if anybody wants to, if you have a passing interest, take a look. But basically, this goes through what we did and what we collected. We ended up collecting 70 species. We ended up collecting 80 specimens. So that's a lot. I mean, we collected 80 actual pieces of algae. And then one of the things that I thought was really neat, and I don't remember any of this, but as we were doing this, we realized we would collect this algae, and then we would try to preserve it as a specimen, and it would just disintegrate. We had to figure out how to preserve our specimens. So there was some experimentation that went on, and we just figured out, we use this chemical and we soak it for this long, and if we soak it for too long, then it decays, so we just had these tips that we put into our paper for future years, and then we made a list of all the things that we collected, so we have a list of the organisms and the places that we collected them, and then there were even pictures, and one of them, this is the map of where our collection sites were, and then here's, there's a picture, so that was my partner, Lisa, and I did remember her, by the way, after I saw this, but I had forgotten about her. She was not part of that sort of core group, but she and I worked together, we got along really well, but she wasn't part of that core group. And then, wait for it, wait till you see this is hilarious. That's me. So, not to belabor the obvious, but first of all, I had hair back then. And you can't tell from this picture, but I looked a lot younger, but otherwise, nothing has changed. I am wearing the exact same clothes, basically, shorts, even to the point where my sweatshirt is sort of 30 years ago, I was the exact same doofus who stands before you today, which I find absolutely hysterical. Anyway, the point of all of this is not, look at how great we were. The point of it is somebody held this out to us as a standard and said, you can do this stuff, you can do this kind of work, go do it, and do it well. And we all did. And what was really crazy about it was that the sense of accomplishment and pride that we got out of doing that. And I had forgotten that we did it, but once I came across all this stuff, I remembered feeling this is a kick butt thing to do. And I had already made a plan for you all. That I was going to do, we were going to do, so we started at the beginning of the year, we talked about ecology, which is kind of a big thing. Then we got small. We talked about macromolecules, the chemicals inside living organisms. And then we talked about cells, and we talked about DNA, and we talked about genetics, and then we talked about evolution. We got bigger and bigger and bigger. And now we're going to go biggest of all and just look at the diversity of all life on this planet. Always knew that's what we were going to do this time of year. And my model for that was a lab activity that a lot of people do something like this. But I discovered it about 10 years ago when I started teaching a class at OSU in the summer. And this is the lab manual for that class. We've changed the class. This is an older lab manual. This is like 6 years old, but the projects, they still do this project, and I don't know how different it is. I don't have the newest lab manual. It might be a little bit different, but it's not dramatically different. But basically, the idea is go out into your community and find living organisms, collect them, preserve them, and learn a little bit about them. Because one of the really cool things about biology is thinking about the diversity of all life on this planet. And so my thought was, ooh, we'll do that. But what occurred to me this weekend as I was looking at our work and being with these people who had been and still are such an important part of my life, what I realized is what I want you all to do isn't a high school modified version of this. I want you to do this. This is an entry-level biology course. This is what freshman and sophomore students at Oregon State University take. And you all are more than capable of doing this work. And you're more than capable of doing it really well. So that's what we're going to do. And what I'm going to do for you is to help, because you aren't college students, you're high school students. What I'm going to do is break it down into chunks. And the chunks are going to be really clear. We'll tackle those chunks day by day. But at the end, your goal is going to be the same as the goal of the students that are going to take the class that I teach this summer, if I end up teaching it, which is you're going to collect specimens from 12 different groups. You're going to preserve them. You're going to gather some information about them. You're going to put them all into a 1-gallon Ziploc bag, and you're going to fork it over toward the end of the last week of school. And then you're going to do one more thing, which is you're going to give a short presentation about one of the organisms that you found. And that is college level stuff. And I'm going to break it down for you, but the expectations are going to be the same. I'm not going to go through all of this today. There's 20 pages of this. I'm not going to go through all this today. I'm going to show you some stuff. You can see some general stuff about how they approach the lab work that they do at OSU. You can see some things like how they expect scientific drawings and observations to look. This is the same stuff I asked you to do in September, right? Remember we went out and drew pictures and I said, don't just scribble. That's not the point. This is your target. This is what actual college students do, and every single one of you can do this with no exceptions. Every single one of you is capable of this level of work. And then the Phyla collection project itself is all described here. I'm not going to read this to you. I am going to, it's on Canvas so you can see. There's going to be rubrics. I'm going to use these rubrics to score you. I'm going to score you using the same scoring guide that they use for 19 and 20-year-old students at Oregon State. And we're going to make cards, specimen cards. So every single one of you, you're going to work potentially with another person if you want, and you're going to make 12 of these cards about every single one of the organisms that you collect. And then you're going to package it all up, and you're going to put it into a Ziploc bag, and you're going to fork it over, and then, hang on, we're going to have a little presentation, and this is what the presentation is going to be like. You're going to pick one of the organisms that you looked at, and you're going to do some research, more in-depth research into it, and then you and your partner are gonna present for 5 minutes about that organism. The list of of groups that we're gonna look at is here. And I'll go into this in more detail in just a second. These are phyla. So the way this document is organized is it's kind of in an outline form. So this isn't a phylum, but these are. This isn't a phylum, this isn't a phylum, but these are. So it's always the farthest to the right level. That is the group that we're looking for. And what you need to do is collect one organism from 12 of these groups. There's a bunch listed here. There's 70 or something like that. There may be more now. This was as of 6 or 8 years ago, but thank you. But for now, just understand that what you're looking for is representatives from any 12 of these groups. Some of them we're not going to have here on campus, and you'll see that in just a second. And many of them you won't even know what they are. You'll look them up and you'll be like, well, that still doesn't mean anything to me. Like some of them you might look at and be like, arthropoda, what's that? And then you look it up and you'll be like, oh, arthropoda, I know what that is. So others, you're just not going to know. You know, if you look up the liverworts, hepatophyta, you're going to see something and you're going to be like, okay, I still don't know what that is. We probably have some liverworts here on campus, but you're not going to be able to find them because you won't be able to identify them, and that's cool. You don't need one of all of them, you just need 12 different ones. But what you can't do is, for example, take 2 organisms from this phylum. Anthophyta is a phylum, you get 1 organism from that, and that's it, right? And Arthropoda is 1 phylum, you get 1 arthropod, and that's it. So we'll come back to that in a minute. One of the things that you'll need to do is figure out, for example, how to preserve your specimens. We're not going to do the collections this week, so you don't have to worry about it today, but this document has all the information you need. So instead of me telling you, I'm just going to refer you to this document, just like they would in the lab at OSU. When you're 19, they're going to say, the instructions are in this document, figure it out. Materials, I will get for you. So if you need, there's chemicals that we need, and we will make sure that, I'll get the chemicals and I'll instruct you on how to take care of them. When it comes to the collection and preservation, one ethical rule in biology is thou shalt not kill unless it's really critical. So we're going to target things that don't need to be killed. And luckily for us, most of the organisms that we see and find are going to be abundant enough that we can collect either part of them, like you can collect part of a plant without killing the whole plant, or we'll be able to find organisms that have already died. Like you could find in any classroom, for example, you could find an insect that is dead already. So we won't have to be killing things, but we will need to preserve them, or they'll just disintegrate. So that's something that we're going to have to think about. So the big picture is that we're going to spend the next 3 weeks on this. We're going to figure out what organisms you're going to collect, and figure out where you're going to collect them. You're going to figure out all the information that's going to go on your specimen cards. You're going to do the collection, you're going to do the presentation, you're going to package everything up, you're going to turn it in, and then you're going to give a presentation. And that will take us all the way to the end of the year. That's the big picture. I know for some of you at least that's probably overwhelming. But what I'm going to ask you to do on a day-by-day basis is going to be much more concise. So what I'm going to ask you to do today is 4 things. And I'll show you some strategies for doing this. The things that I want you to do are find 12 organisms that you think you can collect from Crescent Valley's campus. They need to all be in different phyla. And a phylum is just a level of organization. It's like genus, only it's bigger. There's more organisms in a phylum than there is in a genus. And we talked about taxonomy a billion years ago. It's not important that you know what phylum is specifically, but it's a pretty large category. For example, our phylum is phylum Chordata. And phylum Chordata are organisms that have like a spinal cord or like a sort of predecessor to a spinal cord. That means anything with a backbone is in our group. We're in the same group as chimps and apes and monkeys and also chickens and turtles and fish. Like, that's a big group. There's a big group of organisms that has a spinal cord. We're in that group. And so phylum is a pretty broad category. You're gonna find today, you're gonna just try to figure out 12 things that you think you can reasonably find here on CV's campus. We need to know the species. So it's not enough to just say tree. That's not a species. You do know, though, some trees. We learned them in the fall. And you also know some other organisms because we looked at macroinvertebrates. And we also looked at lichens, which are a little bit complicated because they're not from one phylum, they're from two, because lichens are a fungi and an algal symbiont. So, but there's still examples of at least one of those two groups. So we can like hit with our plants, we can hit at least one phylum. With our macroinvertebrates, we can hit another one. And with lichens, we can hit at least one more, if not two. So that's like, you're already like a quarter or a third of the way there. So you're gonna, and if you just like look up an organism and you're like, okay, seahorse, that isn't gonna work because you don't know that it's here on CV's campus. We might have seahorses, we don't, but we might. But what you really wanna do is like figure out what is likely to be here. So as you come up with an organism from a phylum, I want you to think about why you would expect to find it on campus. And when I say identify your source, what I mean is like, if you talk to me and I say, yep, we have that on campus, you can say Gregar. If you look it up in a book, or another student says, oh yeah, I saw those. Like, how many of you have seen wild turkeys here on Crescent Valley's campus? Raise your hand if you've seen wild turkeys or evidence of wild turkeys. Okay, so now we all know there's wild turkeys here. Your source is other students, but that's pretty reliable. If only one student said there's wild turkeys and it, you know, like it was somebody who didn't know the difference between a bird and a turtle, then maybe you'd be like, well, I don't know if I buy it. But if 4 or 5 students say there's wild turkeys here, it seems like a reasonable conjecture. You might find information online. We have resources here. I've had students collect organisms here on Crescent Valley's campus before, and some of that work is here in the classroom. So we can track down some of these things. Or you might find like a book that shows Crescent Valley being in the range of a particular kind of organism or something online. So, but we want to make sure that it's likely to be found here. And then where on campus it's likely to be found, so that you have at least a target for where you're going to start looking for these things. We're not going to do any collections today, but ideally you'll have a list of 12 organisms that you feel pretty confident that you can find. And this says, feel free to scout if that would be helpful. And what I want you to think about here is, I'm going to set up some guardrails, but just like I was treated at the Friday Harbor Labs 30 years ago, I want you to feel like this is really not me just telling you what to do, like this is you. So the guardrails are, if you walk out of this classroom today and wander around, looking at your phone, talking to your friends, taking selfies, and come back in and you haven't found any organisms to look at, then you wasted your time. I can't police that, but what I'll tell you is, I know you all can find some joy in actually taking this seriously. I know you can, because I can, and I'm not really that different from you. I'm not even that different from me, as you saw from the picture earlier. So if you want, go scout around. Like what I really love more than anything else is if you got excited about something. If you were like, ooh, there's this iris that I know grows in the Willamette Valley. I wonder if we have it here on campus. And you come to me and you're like, Gregar, do we have any of the irises? And I say, yeah, we do. I know we do. They grow over past the tennis courts in the wooded area and along the creek. There's 2, Douglas iris and the Oregon iris, and both of those grow here on our campus. I don't know if you'll find one, but you could go look, and wouldn't that be cool? And so that is what I want, is for you to like actually do some scouting out. Or if you look up an algal group, you find an algae and you're like, algae? We don't have algae here. Wait a second, we have a creek. Are there freshwater algae in the creek? The answer is yeah, there actually are. And so you like go down, scout it out, see what you can find. And then you identify the phylum, and you're going to have 12 different phyla. And I'll show you something real quick that you can do that I think will be really helpful. And then an initial plan for finding it, knowing that it's what you're looking for, collecting it, and preserving it. And we'll see how far you get with all of this today. This is sort of step one. You're not actually going to collect anything today, but ideally, at the end of the day, you'll have a pretty good idea of where things are. Here's how I would go about figuring this out. And it's going to look like it's going to take forever, but if you work with a partner, it actually won't take very long. And some of these things you'll be able to look at and pretty quickly figure out, like what's reasonable and, hang on, I can't see anything. What's reasonable and what's not reasonable for you to look at. So like this group right here, the amoebozoas. Okay, what are those all about? All right, I'm gonna look up, I don't know what this even is. Entamoebas. So I'm just gonna go right here and type, I can't even remember how to spell it. How is it spelled? E-N-T-A amoebas. E-N-T-A. All right, entamoeba. What are entamoebas about? It is a genus of amoebozoa found as internal parasites. Stop. You think we're going to find and collect and identify internal parasites? Nope. Forget that. Not going to happen. What about annelida? You know what that is? Anybody know what annelida is? What's that? Are tapeworms? Is that what you said?
No, like those ones.
Yeah, like worms, yeah. So annelid worms, if you were to look up annelida, what you'll see is, oh hey, an earthworm. You think we can find earthworms here? Yeah. So look, what you can do is just type the name of the phylum in and look. Ideally, what you'll find is some really cool organisms, right? Like there are some that you've already seen that you already know. Oregon ash, you can figure out what phylum that's in, and you could use that one. But ideally, you would look for something that's maybe like that you don't know about, something that interests you, something that you find really fun. And so with like the beginning step, it's go through some of those phyla until you find at least a handful, 12, maybe even more, where you feel like, okay, we've got a fighting chance of finding this particular group, finding something from this particular group. And you don't have to worry about the collection and stuff like that. Down the road, we'll talk about it. Like, so for chordata, you do not need to catch and preserve like a newt. It's enough to use a part of an organism. So I brought up turkeys because turkeys often shed feathers. Turkeys are members of phylum Chordata. That means if you find a turkey feather, you're good. So this is not going to be an exercise in like how do we preserve a whole wild turkey. Like it's not, everything has to fit in a gallon bag, and we're not going to get crazy with the kinds of specimens we collect. But still, it would be kind of fun to target and find things that are especially of interest to you. So your goal for today is just this first piece. Your goal for today is 12 organisms from 12 different phyla. We want the scientific and common names. We want to know some information about where you think you're going to be able to find it, why you think you're going to be able to find it on campus, and some initial plans to like, well, how are you even going to know when you found it, right? Like if you look at some weird plant, will you be able to recognize it if you see it out in the wild? So these are the things that you're going to try to do today. Before we move on, does anybody have any questions about any of that? Yeah.
So do we have to answer those questions for all 12 of them?
Yeah, well, we'll see how far you get. So 2 things that might reassure some of you. Number 1, you can work with a partner, but only one, not groups of 3 or more. If you wanna work by yourself, you can. It's not unmanageable, but it'll be a little bit easier if you work with somebody. And then in terms of like, what is my expectation? I know you can all do this. You can all do a really good job with this. I also know that time is a factor. And so, as always, I'm not gonna ask you to do anything outside of class. So today, if you get 5 organisms and you work your tail off and you're interested and engaged and you're learning, you're good. You do not have to meet you don't have to complete everything in the time we have available. You just have to use the time we have available. Does that make sense? So and that's the same thing we've done all year. I don't want you to feel like you're gonna be penalized for working really hard, but not being able to work quickly enough. Now, if you're looking at your phone and you're horsing around and you go for a walk and you end up like populating your Instagram feed with a bunch of awesome selfies, then that's not the same as working real hard and using your time. But if your energy and effort and time are focused on getting this small task done today, then at the end of the period, when I have you turn in what you've got, you'll be good. Okay. Any other questions? Yeah. So I can help you, but it's if you look at the indentation, it tells you. So for every heading, like look the farthest to the right. So like this group, Episthocons, that's not a phylum because there's this to the right, but that's not a phylum because there's this to the right. But these are phyla because there's nothing to the right of those. This is a phylum. This isn't a phylum, but this is, and 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 this is. And I can help you with all of that too. If you need me to like highlight for you which are phyla and which aren't, I can tell you. But otherwise, like most of the names that you see here are going to be phyla. Okay, other questions? Yeah? No? All right, let's take 5. We come back at 8:56. And if you have other questions, let me know. Otherwise, you can get started, find a partner, and get to work.